Hey guys, welcome back. My last video I sounded pretty tired, and I'm still tired, but I'm going to try to sound less tired. So I apologize for that. Let's jump back into it here. We're going to get into the details of what it takes to transcribe. Transcription is DNA to RNA, and we're going to go through step by step how we do it, because making RNA is very, very important. So first off, we're going to look at a molecule, an enzyme, called RNA polymerase. You might remember DNA polymerase, you've learned about that already. RNA polymerase is going to assemble RNA by linking RNA nucleotides in a chain, and the order of that chain is dictated by the base sequence of the gene. So we copy the gene, only we make an RNA strand of the information in that gene. So the new RNA strand is complementary. So C is bonded with a G in the RNA, and the G in the DNA is bonded with a C in the RNA. A T in the DNA would be bonded with an A in the RNA. And the tricky one, an A in the DNA is bonded with a U in the RNA. So don't let that trip you up. Anyways, we make this complementary strand, and then the DNA, replication and transcription, they both synthesize new molecules by base pairing. So DNA replication is an old thing from last chapter, but the base pairing rule is the same in last chapter as it is in this chapter. And so uracil pairs with adenine, and that's the tricky part for going DNA to RNA, is getting that uracil in there. And the RNA polymerase adds nucleotides to the transcript, so the very important enzyme. Alright, so here you have your original DNA strand. Here's your double-stranded DNA. And C is bonded to G and A is bonded to T, and life is simple. When we want to make RNA, let's say this is our gene. We want to make RNA out of that gene. We're going to open up the DNA, bring RNA polymerase in, and we're going to copy it. G to C, T to A, C to G, A to U. And what we end up getting is the single-stranded RNA, who is a copy of the codes that the DNA had. So RNA polymerase and some regulatory proteins attached to the promoter. Now, the promoter tells RNA polymerase when to start, I guess, where a gene begins. It tells RNA polymerase where to start copying that information from the DNA. So it is a specific binding site very near to the start of a gene that tells RNA polymerase, go ahead and grab onto the DNA here. And so RNA polymerase does that. It moves from a 5' to a 3' direction, just like DNA polymerase did. It unwinds the DNA helix, it reads the sequence, and it joins the RNA nucleotides into a complementary strand, which is a lot of jobs for one enzyme, but it does a pretty good job of it. So that's RNA polymerase. Alright, so here's a cartoon diagram. We got our double-stranded DNA. This region of the DNA is our hypothetical gene. RNA polymerase grabs onto a promoter before the gene is actually here. As the polymerase moves down the DNA, it unwinds the DNA. At the same time, it links these nucleotides for a complementary RNA strand. And after the polymerase passes by, the DNA goes ahead and bonds back together. And that's called the transcription bubble. This thing right here, I guess it looks like a bubble. So there you go, transcription bubble. And then if you zoom in, you can kind of see the RNA polymerase is facilitating these covalent bonds between the complementary bases. And that's how we get this code made. So kind of a cool deal. And then here's the whole thing again. They just made RNA polymerase green. So if you learn it better with green, there's that. Post-transcriptional modifications. So this is a fun topic. You've transcribed DNA into RNA. You are halfway there to making a protein. Do we just send the whole RNA out of the nucleus? I mean, that would be easy. Turns out things are never as simple as we hope. No. We actually do a lot of modification of that RNA transcript before we let it leave the nucleus even. So we have these two pieces of RNA called introns and exons. I remember it this way. Exons exit the nucleus. That might help you. Introns are nucleotide sequences that are removed from new RNA strands. So we literally just built this thing, and now we look at it and we go, this little piece doesn't really belong. This little piece, we don't really need that either. And all the exons bond together, and the exons are the stuff that we will actually send out of the nucleus. Your book compares it to sewing a dress and then snipping off loose material. I don't know if that works for you, great, but there you have it. Exons exit the nucleus, introns stay inside the nucleus. So another thing we do is called alternative splicing. And what this does is it allows one gene to encode for multiple proteins. Very, very cool. I think it's called pleiotropy in our genetics chapter. We'll see. That might be one gene, multiple effects, something like that. So what we end up doing is we remove these exons from our RNA, and we can take other exons and splice them together in different combinations. And depending on the combinations of the exons, we can make a different protein. So once we've done this splicing, we've got a final transcript that's finished. And this is the RNA that we are going to send out. And it has a couple of things that we're going to add on it in this modification process. One is a guanine cap at the 5' end, and that'll help the ribosome find and read it. The guanine cap signifies this is the beginning. And then we have a poly-A tail at the 3' end. So the ribosome will read 5' to 3' too. And when it does, it knows when to start, because the RNA starts guanine, 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 guanine. Okay, that's the beginning. And then we go through and we read these little codons. We make a protein. How do you know when it's done? Well, when it's done, it just goes adenine, 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 on and on and on, like 50 of these things, or 100 or 200. It just goes on and on and on. It's kind of like a skipping record. You know, the same little sound goes over and over and over again, and the ribosome's like, okay, this song is obviously over. All I've read is adenine for days, so it goes ahead and let's go. Cool? So that's how you know how an RNA starts and how it ends. Guanine cap, poly-A tail. So here's what it looks like. DNA, we've got a promoter region, and then we've got our gene. Our gene is made of exons and introns. So we open that DNA up like we did a couple slides ago. We copy it to make an RNA strand. Now we've just got the gene, but we've got some exons and introns. We take those introns out. We just get our exons. Exons, exon, exon, stuck together during RNA processing, still a part of transcription, and we end up getting our guanine cap here, guanine, 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 adenine, 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 and now we have a finished RNA piece that a ribosome can find and read and turn these little pieces into a protein. So very, very cool. We've done a lot of changing in there. So messenger RNA, this is the one who's going to actually carry it out of the nucleus. It's going to turn this thing into a protein, and it's going to go find a ribosome. Ribosomes work with transfer RNA to accomplish translation. The protein building information is carried out in our codons, and a codon is a sequence of 3 nucleotides. That is important to know. Please know that a codon is 3 nucleotides. Very important stuff. Each set of 3 nucleotides codes for a specific amino acid, and the order of the codons dictates the order of the amino acids. The order of the amino acids tells us what the protein's going to be like. And so this is information. It's like 3-letter words, and each little 3-letter word codes for a different word, right? That's what a codon is like. And so that's what we find on our messenger RNA, and those are very important structures. So in our genetic code, there are 64 codons possible. That's in the human genetic code. There's 20 amino acids found in proteins. And so this should kind of seem odd to you. Like, how do we have 64 codons if we're only making 20 amino acids? And the answer to that is called redundancy. We have more than one codon that codes for any particular amino acid. So there might be 3 or 4 that code for leucine, and 3 or 4 different codons that code for isoleucine. And some of these codons will signal the start or the end of genes. For example, the codon AUG, adenine uracil guanine, codes for the amino acid methionine. Methionine is always the start codon. Every protein you're going to make starts with methionine. The others, few that are kind of cool, UAA, uracil adenine adenine, uracil adenine guanine, uracil guanine adenine, these are stop codons. So, how do you know when to stop reading an RNA transcript? You're not only going to hit the poly A tail of adenine, 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 but you're also going to hit a stop codon, which is a codon that does not code for an amino acid. So there's nothing else to hook on the protein. You just let him go, and he's done. It's kind of cool. So this is a fun little thing. I've actually got a lab for you guys called, it's got some weird name like Codon Bingo. It's kind of fun. But what you get is any codon. So let's pick one. Let's make one up in our heads. We're going to make the codon guanine cytosine uracil? I hope that's one. Let's find out, shall we? Okay, so your first base is guanine. Found him. My second base is cytosine. So I go down to guanine, I find my guanine, I find my cytosine. My third base, UCAG, top is U, guanine, cytosine, uracil. That codon that you and I just made up in our noggins there in just a second codes for alanine. Cool. So there's information for an amino acid. And it turns out that not only does GCU code for alanine, so does GCC, and so does GCA. GCG apparently must be a stop codon. Doesn't look like there's much going on there. Anyways, or you know what? Actually, these brackets might just be shifted up a little bit because there's a lot of them that don't have that on them. Yeah, I wonder if it's not all 4 of those. Alanine. That's the answer. Sorry. Anyways, here's your 20 amino acids specified by their genetic code. So, alanine, arginine, asparagine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, on and on and on and on and on it goes. So don't have to memorize these necessarily, but take a second, read through them. They're cool to understand what a few of these are. Phenylalanine, it's an amino acid you'll see labeled on like Diet Coke bottles. It'll have a little message at the bottom of Diet Coke that says phenylketonurics. This product contains phenylalanine because phenylketonurics have a tough time with this amino acid, and it actually causes a lot of problems with their disease. So it's important to know a few of these guys. Tryptophan's one you've probably heard of before. So they're all over the place. 20 of them. So let's see. This is the whole thing pieced together. From DNA to RNA, from RNA to protein. So the region in the DNA that we call the gene, messenger RNA breaks it out into codons. Codon AUG for the start codon, another set of 3, another set of 3, on and on and on. When we translate this, the AUG codes for methionine, the UAC codes for a tyrosine, the UCA codes for a serine, and down the line it goes. So, we're going to leave this right here because the next piece of this is going to be translation. And I said I'd take you through transcription in this video. So we're going to stop right there, DNA to RNA, let you process that. We'll come back next time for translation.